These are your forensic notes for the DNA unit. So what is DNA? DNA stands for deoxyribonucleic acid, and it is the genetic material in our cells. No two people, except identical twins, have the exact same DNA. Although we share a lot of similarities rather. No two people have the exact same DNA except for identical twins. We're going to be spending a lot of time talking about DNA fingerprinting. DNA fingerprinting is also known as DNA profiling. It's a technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. So DNA fingerprinting has been used to help solve crimes by linking suspects to crimes and by identifying bodies of victims. In addition to crime investigations, DNA fingerprinting can also be used to establish paternity and parentage. So if there's a question about if that's actually the father of a child, we can use DNA fingerprinting to solve those types of things. It's also used to identify victims of war and large-scale disasters. For example, it was used in 9/11 to identify victims in the towers. It's also used to study the biodiversity of species. It helps us determine how related different species are. Helps us to track genetically modified crops and to settle immigration disputes. So most lab techniques used for DNA fingerprinting were not intended for these purposes, but instead were developed for use in the medical field for diagnosis and treatment of diseases. As that technology came out, we found that there's a lot of different things that this technology can be used for. So this was not its intended purpose, but it is very helpful in lots of different ways. So where can we find DNA evidence? Small amounts of biological evidence left at crime scenes, which is also called trace evidence, are the source of DNA needed for DNA fingerprinting. So biological evidence includes things like saliva, blood, semen, skin, hair roots. As long as the root is attached to the hair, we can use it. Body tissue cells and even urine will have trace amounts of DNA in it. Because the evidence is capable of identifying a specific person, this is considered individual evidence. There are two main types of DNA. DNA that's found in chromosomes is called nuclear DNA. Nuclear DNA is inherited from both the mother and the father and is virtually identical in all cells of an individual's body. So your DNA is a combination of your mother and your father's. Pretty much all of your cells have the exact same version of that DNA. Mitochondrial DNA is in the form of a circular loop versus a chromosome. And unlike nuclear DNA, it's only inherited from the mother. So using mitochondrial DNA, we can trace back maternal lineages. That would be like just the mother's side of the family, using just mitochondrial DNA. Mitochondrial DNA is passed to offspring in the cytoplasm of the egg cell at fertilization. No mitochondria are going to come from the sperm cell. So both nuclear DNA and mitochondrial DNA can be useful in forensic science. We can use them for different things. We're mostly going to focus on nuclear DNA. The human genome is the total amount of DNA in a cell. It's contained in both the nucleus and mitochondria, so it's all of that DNA put together. That's what makes up your genome. The human genome consists of approximately 3 billion base pairs. The purpose of the genome is to code the blueprint for the human body and how it works. It's responsible for telling the body to make proteins and other needed molecules. It also has the information about where certain things need to be located. It's why your arms are where they are and not a leg. It's all thanks to your genes. Not all 3 billion of these bases are useful code. There are exons, which are encoded by DNA with directions to build molecules. Exons are only about 1.5% of your entire genome. So they code for about 24,000 different genes. The rest of the genome is made of introns. So the vast majority of your genome is what we call introns. Introns are unencoded DNA. They don't code for the production of any proteins. These segments make up about 98.5% of the genome, and sometimes they will be referred to as junk DNA. So despite that name, introns actually have a function in gene splicing and actually have a very important role in biology. And introns are what we're going to focus on in forensic science. So in this whole big segment of DNA, you have the exons, which actually code for something. We have introns, which don't code for anything. They're just filler DNA. Most of the human genome is the same in all humans, but some variation exists among individuals. Most variation in DNA is found in the non-coding segments. It's found in those introns. Much of the non-coding DNA is in the form of repeated base sequences, and sometimes these sequences can be repeated many times. So polymorphisms are the non-coded DNA segments that contain unique patterns of repeated base sequences that are unique to individuals. So those polymorphisms are what make our DNA unique from each other. It's not the actual exons. Those exons are actually very, very similar in every person. The introns are what make each person unique. So obviously that's the part that we're interested in when we're doing identification. So DNA fingerprints are created using a process called gel electrophoresis. The result is a series of bands representing fragments of DNA. Fragments are created with introns from the genome. So you can see here we have 9 different wells or 9 columns, and each of these little stripes you see, we refer to those as bands. And each person will have a unique set of bands because their introns are going to be unique. Intron data from DNA fingerprints can be analyzed for 2 main purposes. So the first purpose is inheritance matching. So this is comparing family members' DNA for proof of familial relationships. So we can look at a child, a father, and a mother. Every band in the child's DNA fingerprint must be present in at least one of the parent. Because you get half your DNA from your mom and half from your dad, all of the bands, so to speak, in the DNA fingerprint of a child have to match to one or the other. For example, in this one, here's the mother, here's the father, here's the child. It matches this one. You can see these faint bands here match the father. This one up here matches the mother. Same here. This matches the mother. This matches the father. So every band is accounted for every band is either going to match the mother or the father. So we would say this is a positive match. So every band from the child is going to match one or the other. Tissue matching is what we use often for forensics. It's comparing DNA evidence from a crime scene with a DNA sample taken from a suspect. If the two samples have the same pattern, that means they're from the same person. So we're going to collect DNA from the crime scene. We're going to take cheek swabs from different suspects or blood samples. It can be done either way. And we're going to see, do any of the suspects match that DNA at the crime scene? So in this case, the crime scene matches suspect number 2. Number 1 and number 3 do not match what we found at the crime scene. Often the amount of evidence left at a crime scene is very, very small and therefore considered to be trace evidence. The problem with analyzing trace evidence is that many forensics tests are going to destroy the evidence sample, as is the case with DNA. When we run a DNA fingerprint, when we run it through a gel, that DNA cannot be extracted and used for any other tests. So before we do DNA fingerprinting, we do something called PCR. PCR stands for polymerase chain reaction. PCR is a technique that makes thousands and thousands of copies of segments of DNA that investigators want to analyze. The DNA produced by PCR can then be used to make a DNA fingerprint without worry of destroying the actual evidence that was collected. A large amount of blood is needed to create a DNA fingerprint, but only a small amount is needed to use PCR and create many copies. So you can see here, this large blood stain, that's how much we need to run a good DNA fingerprint. That tiny little drop there is all that's necessary for us to do PCR. PCR is basically like a Xerox machine, right? It's like a copy machine for the DNA. It's going to make many, many, many copies. So that way we can save some of it, preserve some of it for future testing if needed, and we can use those copies to run the DNA fingerprint. So here are the steps of creating a DNA fingerprint. First, we need to extract the DNA. We're going to isolate the cells from the tissue, and then we're going to disrupt the cells, basically destroy the cell membrane in order to release that DNA. Then we're going to cut the DNA into what we call restriction fragments. Because DNA contains long strands of genetic information, they need to be cut into smaller pieces. We're going to cut them using restriction enzymes. Those restriction enzymes are sometimes called like molecular scissors. They cut DNA at a very specific location. So after we add that restriction enzyme, the DNA contains smaller fragments of all these different lengths. There's a lot of different types of restriction enzymes, but the way they work is that they read the DNA code looking for a specific sequence, and then they're going to cut the DNA at that location. For example, this particular restriction enzyme looks for the sequence, excuse me, AATT. When it finds AATT, it's going to cut at the beginning of the A, all the way down, and then at the beginning of the A on the opposite strand. So then we have now 2 pieces. And every single time it sees this pattern, the AATT, it's going to cut the DNA in that location. Next, we're going to do a process called, or a step called amplification. This is basically making many copies of those fragments that we just created. So again, we're going to use PCR, polymerase chain reaction, to create many copies of those fragments. Then electrophoresis is the process that separates those fragments according to their length and ultimately creates our DNA fingerprint. So this is what that looks like. All right, we load the DNA in one side into these wells, we call them, into this gel agarose layer. So we load the DNA into these wells, one sample in each well. Then we attach it to some electricity. DNA has a slightly negative charge, so it's going to be attracted to the positive end. So as we run electricity through the gel, those fragments are going to be moved towards the positive end of the gel. They're going to be separated by length. Heavier, longer fragments are not going to travel very far. Shorter fragments are going to travel all the way to the end of the gel. And that's how we get those different bands. When we're doing a DNA fingerprint, one of the wells should contain a control solution, meaning the DNA fragments are of a known length. This is called a marker or a standard DNA. You can see here, that's what this one is here. We have 3 different sizing markers. This was basically just a sample where we know exactly how long the DNA fragments are. Then we can compare it to our samples to figure out how many base pairs each fragment contains. The other wells should contain DNA from the crime scene, from the victim, and from the suspect. So you can see here we have victim, suspect 1, suspect 2, some sperm, and the female, right? So we always want to test the victim because we want to make sure any blood, for example, at the scene isn't from the victim. We're looking for the suspect. So we always run the victim, then any potential suspects, and then any of our DNA samples from the crime scene. So in this particular one, we have 1, 2, 3, 4, 5 different samples that we're testing, plus the control. So at the end, the DNA fingerprint is going to look like a striped column. In order for DNA fingerprints to match, the columns must have bands with the exact same places, with the exact same widths. So we should have the same quantity of DNA in the same location. You can see here, like these are 2 very thick bands. These are 2 very thin bands. We want to match both the thickness and the location. Once we have that DNA fingerprint, we can match crime scene DNA with the suspect. It also can help us to eliminate a suspect, right? If their DNA doesn't match, that is evidence that it isn't that person, right? It can also be used to free any falsely imprisoned individuals. If someone is convicted of a crime, let's say in the 70s, before DNA technology was used, we can use fingerprinting now to prove that that person didn't commit the crime. We can also use that DNA fingerprint to determine maternity, paternity, or to match to even other relatives. We'll talk a little bit about DNA genealogy, where they're making family trees based on DNA testing. And this can also be used to identify any human remains. If the remains are not able to be identified by other means, we can use the DNA fingerprint to match with maybe something we have on record from them, or a sample we can collect from like a hairbrush or a toothbrush. That way we can positively identify them. So how do we collect the DNA evidence? Because DNA is found in all cellular material and cells are very small, contamination is a very important issue to address when collecting and preserving DNA evidence. To avoid DNA evidence contamination, crime scene investigators should make sure they wear disposable gloves and change them often. Between every sample, you don't want to cross-contaminate the DNA. Use disposable instruments for handling each sample. For example, if you're using tweezers or forceps, you want to use a different pair for each sample. You want to avoid talking, coughing, or sneezing over evidence, because any time that you have saliva leaving your mouth, DNA is going to be contained within that. You do not touch your face or your body when collecting or packaging evidence. If you're, scratching your eye and then touching evidence, you can transfer your own DNA. We're going to air dry the evidence before packaging. If the evidence can't be dried, then we can also freeze it. That's in order to protect that DNA, prevent it from molding and being destroyed. We want to avoid using plastic bags to store evidence that contains DNA. We're going to use paper bags or envelopes. We want to keep evidence in a cool and dry during transportation and storage, avoiding direct sunlight because that can actually damage the DNA. Where do we store this DNA evidence information once it's collected? Each state keeps DNA profiles of individuals who've been convicted of certain kinds of crimes, such as rape, murder, and child abuse. The military also has DNA profiles of all servicemen and women. And the United States electronic database of DNA profiles is called CODIS, which stands for Combined DNA Index System. DNA profiles from crime scene can be entered into CODIS to identify any possible suspects that may match. DNA profiles are so unique that there is a 1 in quadrillion possibility of a match, right? If it matches, there's basically no way it can belong to anyone except that match. As of February 2011, so that's 10 years ago already, CODIS contained 9.4 million profiles from DNA of convicted felons of violent crimes. Of 360,000 profiles from DNA at crime scenes, CODIS was able to match 138,700 hits, is what we call them. Hits are what we call them matches. So I'm sure that number is much bigger today since it is 2021. Genetic genealogy can help to create new leads on cold cases and help identify what we call Jane or John Does. Those are like unknown victims of crimes. Direct-to-consumer tests from companies like 23andMe or Ancestry.com can create genetic genealogy databases. It allows genealogists to find relatives of a suspect by comparing the databases to DNA left at a crime scene. A very famous example of this is the Golden State Killer. He was wanted for at least 12 murders and 50 rapes that happened in a 10-year span between 1970 and 1980. They discovered that his name is Joseph DeAngelo. He was identified as the Golden State Killer in 2018 thanks to the public genetic genealogy database called GEDmatch. So anytime that you submit your DNA to a private company for analysis, you have the option of also submitting that information to a public database called GEDmatch. GEDmatch is available for investigators to use. By submitting it, you give your consent that they're allowed to look at it. And essentially, they were able to trace the family tree of the Golden State Killer based on DNA that was left at his crime scene, narrowed it down to a couple people all within the same family, and then found that Joseph DeAngelo matched the location, the dates of all the crimes they investigated, gathered more DNA, and they found a positive match. All right, so that's the end of your DNA notes for forensics. Please make sure you've completed all of the blanks and turn it in on Canvas.